everybody. This is great to see such a crowd. This is awesome. Welcome to the Department of Biological Sciences seminar series. We've got certainly a special seminar today. Really happy to introduce today's speaker. If I had known that some of his former graduate students were going to be here, I probably would have asked them to do it. Sorry, Jason, etc. But
Thank you, Nathan.
You know, if you guys did it, Wynn wouldn't have nearly as much time to talk. So, really happy to introduce Dr. Wynn Watson today. As you might infer by the title of his talk, he's been here for quite a while. His first year here at UNH was the 1978-79 academic year, so this is exactly 40 years, which is incredible. Wynn grew up in Connecticut, did his bachelor's at Wesleyan University, also in Connecticut, and then went on to do his PhD at UMass Amherst in zoology. He finished that up in 1978.
After I started working here.
Yes.
Good luck with that stunt these days.
Immediately came here in 78 and has worked himself up through the ranks. He is a marine biologist, marine ecologist, marine neurophysiologist, and also I would say a behavioral ecologist as well. So he wears many hats using invertebrates as his model systems. He has written, he didn't actually number his publications, so I had the pleasure of actually trying to count them. He has over 90 peer-reviewed publications to his name. But really, when I chatted to Wynn about introducing him, the things that he wanted me to mention was in terms of how many students he has mentored. So 25 master's students that have come through and graduated through his lab, an additional 7 PhD students. And maybe the most important metric of all, he even told me once that this is how you measure your value as a faculty member, is how many of your previous students' weddings you get invited to. To which he estimates that at approximately 15. Certainly his mentoring has been noticed, not only by his students, but by the university as well. In this year, or just a couple weeks ago, he was awarded the UNH Graduate Student Mentor Award. And a few years ago, in 2013, he won the UNH Award for Excellence in Teaching. So certainly a valuable member of our department, so I'm really excited that so many folks came to learn some lessons from him. I'm certainly looking forward to learning more from him. So take it away.
I thought he was going to say, I'm very excited to see him leave. Because he's getting half of my junk when I leave. Thank you, Nathan. So today, I'm going to talk to you about the various creatures I've worked on, right? And I got to go in semi-chronological order, but you know, I try to mix up chronological order with animals. It's just not going to work. So let's go way back. And so I interviewed here in 1978 in the spring, and this was a lot of the zoology department at the time. And I made this picture up for John Saster, my first mentor here, and because he actually played on the team, and that is actually John Saster.
But he was a faculty member of the team.
Yes, sir. Your emeritus basketball player. And these guys were crazy enough to hire me, and the rest is history. So I do want to start by saying that there are a lot of people, whose work I'm not going to be able to mention. There's some piece down here, guys. Unless you want to stand. There's a lot of people who did some great work in my lab, but I don't have time to mention everybody's stuff, so I apologize to those people, especially if you're here. So I'm going to go back to my roots. I'm going to go back 450 million years to these guys, right? So here's your basic horseshoe crab, right? And when I went to graduate school, I worked on these guys, and I worked on how they breathe. And in case you don't know, these are their gills here, and under the gills are these pages, like the pages of a book, and blood flows through the pages, and they flap their gills. in order to breathe, right? And so I was working on that breathing rhythm, right? And it looked kind of like this if you film it. So you could, you're hearing the muscle that closes the gills fire, which is this guy. Okay, so they're flapping the gills. So you can take the nervous system out of these guys, and after you've identified the muscle activity that occurs while they're breathing, you can take out their nerve cord, you can put it in a dish, you can record from the nerves that go to the various muscles, and lo and behold, they continue to breathe when the nerve cord is sitting in the dish. So this is what we call a fixed action pattern that's produced by a central pattern generator. No sensory inputs required, it just does it automatically, right? So life is good. And then one night I'm working in the lab, and I look over, and I should back up and say, sometimes it doesn't look like this. Sometimes it's really messy, and I had no idea what was going on. So I'm sitting there in that lab one night at night, and I look over, and I see a guy doing this thing. Right? It's like chaos. And what they're doing is they're cleaning their gills. They're taking one gill, and they're sliding it between the pages of the other book on the other side. So this is called, I called it, gill cleaning. And lo and behold, if you take out the ventral nerve cord and put it in a dish, they will clean and breathe and clean and breathe for hours. And this is one of the most complex central pattern generators that's ever been identified. So this guy is not only breathing, that's easy, but he's cleaning. Kind of a crazy thing. And then they alternate different kinds of cleaning because you got to clean one side then the other. And it's made into a textbook, lo and behold. So then I turned to these guys. Why did I turn to these guys? I wanted to figure out how that cleaning thing worked. I wanted to know how the breathing thing worked. But of course, you could have nerves aren't very big. But this is a nudibranch, Melody leonina, and this is its brain. And each one of these round things is a nerve. So they're really easy to work on. These gastropods have the biggest neurons in the animal kingdom. So you can put this thing in a dish and see the nerves and start figuring out how things work. And I was kind of obsessed with figuring out how you wire together neurons to create a rhythm. Okay? And so first, I had to invent a contraption. Okay? So this contraption allows, see this guy swimming right here? I'm recording intracellularly from a motor neuron involved in swimming while he's swimming. Not easy to do. So you have to kind of put a platform under the brain, pin the brain to the platform, put an electrode in there, and hold everything steady while he's doing this. Right? But as a result of doing that, you can begin to identify some of the cells involved in swimming, right? And then when you take the brain and put it in a dish, you can find those same cells, and then you can do some circuit work. So I ended up, you're not supposed to understand this real well. This is the neural circuit. All the neurons involved, except for some new guys, that are involved in creating the swimming patterns. So for example, you can see when this guy fires, he inhibits the guy on the other side. And he excites the motor neuron on that side. So go back and forth, right? So we invented this, we invented this tool. We identified this circuit. There aren't a lot of circuits out there for which you know all the neurons. While we're doing this work, we discovered, sort of accidentally, that they swim more at night. And then I went out to the gym to figure out the day to actually document that, yes, they're nocturnal, they swim more at night. There's a circadian rhythm that's powered by circadian clocks. And now we're still trying to figure out how that clock talks to the circuit. But we have all the pieces. We know the circuit, and we're honing in on the clock. And so now we can figure out how is it that circadian clocks modulate when you do certain behaviors. So that's going to haunt me the rest of my life because I'm retiring, but maybe you can pick up where I left off. Okay, so now I'm going to shift gears. I'm going back in time again. Right? So way back, some people reporting a decline in the number of salmon in rivers around the world that were moderately acidified, but not so much as to kill salmon, right? And so you all know salmon find their way up rivers by smelling their way up back to their home stream. So we hypothesized that maybe the drop in pH due to acid rain affected their sense of smell. So Carl Mulligan came to work for me, and we did a simple study This is a Y maze with a fish in it. We drip in some glycine. They like it. We change the pH from 7.6 to 5.1. They avoid it. We change the pH back. They like it. Okay. Cool. Most importantly, and shout out to John, okay, we had to invent this technique for doing this, right? This is, this is way back. We had no way of automatically tracking the movements of fish, right? So we created this system and we published it because no one had ever done it before. And it was really high powered. I mean, the intensity of this computer, as you can see, Apple 128K memory, and importantly, 2 disk drives. One for the software and the other for the data, right? Because one disk drive couldn't hold all of that stuff, right? And so we invented this, and it was based on a system that Kate's dad helped me develop a little bit before this, to track. So more recently, with the ocean acidification phenomenon going on, we became interested in whether this could interfere with the sense of smell of ocean species, right? And so we've been doing this work for a little while. This is work that Stephanie Sykes did. It's at the Coastal Lab. Lobster hangs out at this end in a shelter. We place food behind this barrier down at that end. These are 2 separate channels, and we can change the pH and so on and so forth. And we found that it took them a longer period of time to reach the food at a low pH versus a regular pH. So it seemed to be interfering with their sense of smell, kind of like the fish. More recently, Nathan and Ben have been working on juvenile lobsters down in the G31 area. And this is kind of what it looks like. The normal pH is on the left, the low pH is on the right. And you'll see Nathan stick his hand in and drop a black container with food in it. And then you're going to see that the guy on the left kind of starts to pay attention to it, and the guy on the right does not. So again, this is, we have some pretty good data, but we haven't finished this work, suggesting that when you change the pH, it sort of, we think it interferes with the ability of the odorant to bind to the olfactory receptors, and therefore it either changes the sense of smell or decreases their sensitivity or something like that. So this is something we're very intrigued with, both because it will interfere with their ability to find prey and bait, but that's a whole other thing. But they also use pheromones for mating. And so if they can't smell the pheromones, that could be an issue. Thanks for being patient. If you have an urgent question, because I'm jumping through different stuff, go ahead and raise your hand. If it's a lousy question, I'm not going to answer it. But if it's a good question, because I'll be jumping around here. Okay, so again, I don't know how long ago, but some time ago, Chris Shabbat, who works at Plymouth State, spent his sabbatical here, and he had a background in biological clocks. And so he thought, why don't we do some sort of clock thing with horseshoe crabs? And at that time, we knew that horseshoe crabs mated at high tides in the spring. And we didn't know whether or not they knew it was high tide because they had an internal clock that told them that, or whether they just felt the tides change. And when the tide got high, they went to shore, right? And so we decided to do this study. Wait a minute. Let me make sure I haven't skipped anything. So we decided to do this study. And to do this study, to test if they had an endogenous tidal clock, we had to invent the horseshoe crab running wheel. And so horseshoe crabs get inside that wheel, which we made out of one of these guys. Okay? And there's a magnet on there, and every time the wheel turns, it clicks the magnet, and it records the data, which looks kind of like this. Each tick is a rotation. These are 35-day periods, right? So it can record from these guys in a garbage can in the lab for days at a time. Obviously, there's no tides, but as you know, the tides, you get 2 tides a day, and the tides drift a little bit every day, and these are perfect tidal rhythms. So these guys are expressing perfect tidal rhythms in the lab in a garbage can. Clearly, they have an endogenous clock. Okay. They also, you can synchronize the tides by changing the depth of the water, so they can't feel the depth, but they have an endogenous clock. And we also could put an eye cap on them while we did this, and flash lights to show that they also have a circadian rhythm of visual sensitivity. So at night, they can see better than during the day. 2 different clocks, one in. So they come up at every high tide to mate. We noticed that they seem to be releasing some stuff when they were doing this. You oftentimes see a dozen males or something surrounding one female. We thought maybe they're releasing some sort of pheromone. So we came up with this highly sophisticated technique for testing that. You're gonna put a female in here, find a female that's laying a nest, put a box over her with holes, see if males, surround her. And so we do this every high tide. Sometimes, most of the time I'm there by myself, and oftentimes I forgot my notebook and ended up writing down the data on like a 2x4 or something like that I found in the woods. And then the semester starts and this guy comes on my lab, is there anything I can do? No, get away. Is there anything I can do? Here, take these 2x4s and transcribe this data and let's see what we can find. And that was Steve Jury, who's been collaborating with me ever since. He worked with me as an undergraduate, but I kicked him out, and then he came back to haunt me as a PhD student, and now he's still hanging around doing work. So we demonstrated, yes, in fact, these guys are releasing a pheromone. And then recently I was reading some papers about the Japanese worship crabs. Turns out they're pretty sure tetrodotoxin is the pheromone that they release, and it protects their eggs from getting eaten too. So then, like 2 days ago, I got a call from this girl in Sweden. Hey, I'm interested in pheromone. Can I send you some samples? Can you send me some samples? Yes. So I got to check for that stuff. But that would be kind of cool, right? And speaking of toxins, very early in my career, I was working with John and Tom, and there's this undergraduate kid I met in intro bio, named Toshi Hoshi, and he's a freaking genius like I've never seen before. And so Toshi comes by my lab, same thing, can I work? No, okay, take this and read it. It was like the book on neurophysiology. And he came back the next day and he knew all the equations and the whole thing. Okay, he's now like head of the Neuroscience Institute. So Toshi came in and he got all these recordings, recordings from a crayfish, and demonstrated that this toxin that's in these blue-green algae was just like TTX and Sachs toxin. It blocked the voltage-sensitive sodium channels responsible for conduction of neural impulses, and that's why it's toxic. And so that was a fun collaboration. Sometimes you do studies because you write a grant and you get the money. Sometimes you want to do something, but you have no money. But you kind of figure out a way to do it. And that's what this study was. And this was also done by a UNH undergrad, Heidi. And we were trying to figure out if lobsters make sounds. And so looking here, it's the lobster vibrating its shell, and we're picking it up with that accelerometer. Okay? So it doesn't take a lot of effort to do this, a lot of money. So I did this as a graduate student and published a paper on it. And it was a fun, really fun piece of work. But we had no idea, nonetheless, why they do this. Like, why are you making the sound? Are you communicating with other lobsters? But males make them, females make them. So we didn't think it was a mating thing. And a couple of years later, we did this experiment. So there's a fish in the tank of a lobster, it's a cod. That's the sound of the lobster. And you can see as soon as you did it, the fish swam away.
How was that?
And, we also found out during the same, so we thought maybe it's some other chemical stimulus or stuff. So we bought this on eBay. And we would dangle it in the tank and pretend it was a fish, and lo and behold, when you brought it close to the lobster, it would make the buzzing sound. Right? So try to convince the BSc that this is for science, not easy. Oh, by the way, anybody know what this is? Okay. You saw those, this is old school. That's how you get these. You have to get them on a oscilloscope, take a picture, develop it. Right? Obviously, you can do a very long experiment.
There's a frame rate on that?
We made it one.
And yes, it hurts.
So again, we published a paper showing that these sounds seem to be aversive to predators. We also did an experiment where we had some striped bass in there, and after they encountered the sounds a couple of times, they avoided the lobsters completely. So it also, the predators learn to stay away from this sound, however it's bad for them. Right? We still have not succeeded in recording from any sounds in their natural habitat. We hope to be able to do that soon using a device I'll introduce you to later. Okay, so again, about I'm in the middle of the lecture. Come on, man. No. Yeah, it's okay. You didn't have to come. Yes, I'll give you some credit for this old stuff yeah, I'll give you credit for that. I'm not talking about the aquaculture thing. Okay. All right, okay. Sorry about that. So we got these calls from people who were curious about lobsters in the Great Bay Estuary right after a big hurricane. And I'm trying to explain this, but I can't. So I look it up, and there's nothing. Right? So we started working on horseshoe crabs. Hunt and I started working on horseshoe crabs. Lobsters in the estuary. Did I say horseshoe crabs? Lobsters in the estuary. And one of the things we want to find out is if they can sense temperature and salinity, and then maybe react to a storm by moving. Right? And so we invented this thing here, this lobster cardiac assay, which is going at strikingly low speed. Never mind. So lobster heart rate, when it detects anything different.
There it goes. There it goes.
Oh, beautiful. When it, like I just touched it, it's heart will stop. So it's a startle response. Almost all animals have it. And we thought we'd use this as a way to talk to the lobster. Like, did you sense something different? Okay, so it's in a tank by itself in the dark, other than when I film it like this. And then we're going to add a little bit of low salinity water to see if it can detect that. You'll see a little bluish water come in because I dyed it just for the video. And when this starts to happen, you'll see shortly thereafter, this heart will stop as well. Right? And so we use this kind of a thing. I wave that in front of the thing to let you know that it's happening. You see the blue water come up, heart stops. And you let it go for a little while longer, and you start getting uptight and start to move, right? So one signal tells you it can sense it. The moving tells you it doesn't like it. So we showed it can detect very small drops in salinity. Then we said to ourselves, what about temperature? Because temperature is more universal. Like salinity is only an estuary relevant thing. And so then Steve and Glenn and I did this experiment with a little bit of help from them. And this is a large tank, bigger than this table, in which we create a gradient from hot to cold. Put the lobsters in there, put a logger on them, and see where they go. They love 16 degrees centigrade. They won't go much warmer than 23 degrees centigrade. They won't go cold. Okay? So they do what we call behavioral thermoregulation. They seek water at a temperature that they prefer. And importantly, they avoid warm water. Okay? So we also couldn't help ourselves. And we were wondering about this whole thing, and we invented this guy. Right? So this is the lobster treadmill, right? Not the shrimp treadmill you've seen on Letterman. And the treadmill rotating is on the bottom. It's, when he walks on it, it rotates, and his heart rate's on the top. So we use this to further our studies of their response to salinity and show that at some point, like in the other tank, they'll find it aversive enough to try to avoid it. So at one point they can sense it, at the other point, another time they walk away from it. And so these responses, their sensitivity to temperature and salinity drives their migrations, especially seasonal migrations. Both horseshoe crabs and lobsters tend to move offshore, or down estuary in the fall, and then back up because they're seeking warm water in the spring and early summer, right? And in case of the estuary and some other places, this can lead to skewed sex ratios. Like most of the lobsters in the upper reaches of the estuary are male, and down out of the shoals they're female. Right? And importantly, now climate change is having a big impact. Okay, so these are landings. Georges Bank, southern New England's crashed. Gulf of Maine were doing well because their temperature is still appropriate, right? But the lobsters are sort of disappearing in Long Island Sound, Massachusetts, and so on and so forth. And almost everybody agrees that temperature has a lot to do with it. It might be driving their migrations, it might be affecting other things. So this got us thinking, well, if they're doing these big scale migrations, what about small scale? So we did this study. That's the movement of a lobster in a mesocosm that's out near the coastal lab. Here's a diagram of the mesocosm. We have this telemetry system that we use to track these movements. We had to build a corral about half the size of a football field outlined in here. They did not move outside of it. That was bad on my part. And so you can see, what's this lobster doing? It's leaving its shelter, it's wandering around, and it's coming back to its shelter. So they're capable of homing behavior. And this is, again, this is in pretty blah habitat, sometimes at night, in an area that you know, we sometimes get lost in. Here's this is just a guy walking around down there with a finger on it. And again, there's not a lot of landmarks down here. So these guys are somehow finding their way around within this enclosure.
What's the time scale on the track on the right there?
Like every dot's about 5 minutes. So this is like one night in the life of this lobster. So they're capable of these homing, but like every animal that does homing behavior, you got to ask how do they know where they're going? Right? It's a very fundamental question. And we tried a lot of things. But this is my favorite contraption from the study, right? So you take a tube, you have it connected from the top, from the surface down to the bottom. You have a camera mounted at the bottom. It goes to a buoy that transmits the video back to the boat. You sit on the boat and you drop lobsters off the boat and down the tube, and then you use the camera to watch where they go. Right? And so we did this, and we did this with their eyes covered and so on and so forth. We still don't know how we do it. We do know that they can orient well with their eyes covered, so it's not a visual thing, and they do run around at night. We don't know if it's a magnetic thing or a wave search thing or what the cues are.
Did you cut the antennas off?
And we cut the antennas off and it had an impact. And I'm dying because they just did a study, as the kids in my class know, right, where they cut the antenna off of butterflies and they squeezed them up. And they just found magnetoreceptors in butterfly antenna. And they just found a circadian clock in butterfly antenna. So I'm dying to do that. I got work to do. Okay. By the way, at this point, I should mention, give a shout out to these guys. Virtually everything I've ever done in my lab has been involved these three components. Right here, talk to you. Okay, so they're walking around at night. So we're trying to see if they have a circadian rhythm, like the horseshoe crabs have a circadian rhythm of visual sensitivity. So Steve and I built this contraption, and it's a racetrack. And as they walk around the racetrack, there's sensors in each of these little tunnels that record their movements. And you can see under normal conditions, they will be active at night. But then when you make it dark, that rhythm drifts. That shows it's an endogenous rhythm. Right, they have an endogenous clock that tells them when nighttime is. They can synchronize to the day with their endogenous clock. And when we built this contraption to measure the visual sensitivity of the eye, their eyes are less sensitive during the day than they are at night, a lot like the horseshoe crabs. So they can see better during the night than not better, they have improved sensitivity to light at night. And then recently, we have been doing experiments to try to figure out if they can sense light without their eyes. And so we cover their eyes. I don't know if you can see at the top, but their eyes have this big black thing over them. And then we use that same cardiac assay, and we flash light on them, and you can see their heart stops. Okay? Their eyes are covered. And they sense the light. And if you believe me, so this guy, with flashlight, you can see he gets startled, he moves around. Okay? So they have photoreceptors someplace other than their eyes that they can use to sense light, and I don't know why.
How long does their heart stop?
That's not long. That's like
So it's not.
that's seconds. Right. Not, sometimes it's just like 3 seconds. Sometimes it's like 10 seconds, but not for long.
Longest I've seen is about 2 minutes.
So it's more like skip.
More like a skip beat. Do you know when you see the blue lights flying with that? That kind of. Okay, I'd be remiss without talking about sex. Over here you see a female lobster molting when she comes out of her shell and she's very soft. They do that about once a year. This is also the only time when they can mate, when the female is soft. And so what happens is the female approaches the male who owns this shelter. And then the female starts and the male starts releasing some pheromones, right? And that sort of signals everybody it's good. And she gets into the shelter, and one of the things the pheromone kind of does is make the male passive so he doesn't eat her when she molts. Because if you cut off his antenna, he does. So it's so there's an important pheromone involved in this process, right? And so we were also at the same time sampling females out in the natural habitat to see how many of them mated. We were worried because they protect females over males. There weren't enough males to go around, not enough sperm to go around, so on and so forth. When we did that sampling, we discovered that there was a lot of females that seemed to be immature that had sperm, like they had mated, like what's going on here. So we did this laboratory study where we could actually watch and confirm, and we were able to take sperm samples from the females, right? And that's what a spermatophore looks like over there. And we were able to also cut a little window in her shell and reach in and see if she had immature or mature eggs. And lo and behold, 11 of 14 of the immature females we looked at had mated. So these immature females that are not capable of using the sperm they get to fertilize their eggs because their eggs aren't ready were mating. So we think they do this to trick the males into letting them come into their shelter and molt and protect them while during that vulnerable time while they're soft. And so when I was interviewing for my job here, like halfway through my interview, my girlfriend walked in the room. There she is. As she just did. But she wasn't pregnant with a baby at the time.
Is she now?
No, she hasn't. I shouldn't say she wasn't with a child at the time.
Now look what we've done.
Well, we're clapping. Luke, it's okay. It's okay.
Doesn't want you to retire either.
Right. So here's one of our best contraptions. It's called Lobster Trap Video. For reasons I don't have time to tell you, we wanted to figure out what was going on with lobster traps and were they giving us a good index of what was actually on the bottom. And so, again, this is long ago. And so we had to take a regular VHS machine and put it in this case, fill it with batteries, all these other electronics, and then put it in this trap. And you can see, it took 4 of us to pull this thing out. It was heavy, okay? And so we captured these old school videos of these lobsters in and around traps. Okay, and in this picture, the entrances are right here. Okay, and you'll see that guy's eating bait, and so the guy comes in and says, "No, give me the bait." Right, and then he leaves. See how hard it was for him to get out of that trap? And he comes in the other side. Right? And then this other guy who only has 1 claw thinks, "I better leave because this guy has 2 claws." Right? Not very impressive traps. Right? And so we found that lobster traps are not, in fact, very good traps. And not only can they get in and out, but they chase each other away. So we catch about 6% of the lobsters that actually enter the trap, and only 1 of 5 that approach ever go in. so they're not very good traps. They're not very good at telling what's on the bottom. But they are when we're using the fishery statistics because they aren't as efficient. And this stuff has had a little impact on how we manage the fishery, because now a lot of state agencies use traps without escape vent to try to increase their catch and therefore get a better feeling of what's on the bottom. And then I'm going to jump ahead. So one of the things I'm always doing is studying behavior, activity, rhythms, and I had to use video and all kinds of other odd techniques. But recently, the accelerometer technology has improved considerably. So now we use those a lot. We can either use these hobos in the laboratory or in the field. We can put transmitters. There's a transmitter on this guy. And so we can study their rhythms either in the field or in the lab. And this is a horseshoe crab rhythm recorded not in the lab like the one before, but in the field. So they express good tidal rhythms in their natural habitat. All right, so these accelerometers are great. And then I'm jealous. Recently, Dan invented this thing, the heart and activity tracker. Inside of that little canister is a little microcomputer, and it's capable of recording compass heading, activity, accelerometers, or activity, here's the x-axis, and total activity, and then heart rate. So you can record a lot of the same things you can do in the laboratory now in the field, right? And you combine it with some tracking data, and now you can start asking some really intriguing questions. Like, Ben just got a grant to figure out when do they feed by putting little sensors on their feeding appendages. And I'm trying to convince them to put a little sensor on their shell so I can find out for once, do they ever make sounds in their natural habitat? Right? I mean, don't you think we should figure that out, man?
It's just a really high noise to signal ratio on that one when we've been doing this.
So we're working on that. But the best contribution to date, 4,000 students. I estimate I've taught in my lectures and labs. About 200 undergraduates in my research lab, a bunch of graduate students as well. And so my feeling is if you want to change the world, you create like all those people who can do it for you. Right? And send them out into the world and do good things. And if you're successful, like this year's group, they all get jobs. I don't know how they do it, but I'm finding a hard time finding people working this summer because all these students got jobs.
I didn't.
Dan does have a part-time job this summer with me. If he builds a sensor. So
I'll add it to the pile.
it's been a good trip. It's been a good trip, lots of fun. From here, there's some machine.
It's still up in the Redmond penthouse.
Pardon?
That one's up in the
Back
penthouse.
This was when Susan Alcott, one of my grad students, tricked me into thinking it was a 60s party at the bowling alley. I'm the only guy who showed up like this. They're all like, "Got you." Okay, that's the real end. Questions, Tom?
So with the limulus wave work, and it relates to some of your other studies, did you look at really young animals? In other words, was the exposure to many, many wave cycles imprinting them?
Very good question. Yes, Chris Shabbat's doing that right now, and if you take animals from Florida where there are hardly any tides, they hardly have any rhythms, but if you expose them to tides, so they will train to it, right? And so now what he's doing is he's taking eggs from Florida crabs that don't have much of a rhythm because there's hardly any tide, and bringing them up here and exposing them to tides, either one tide or two tide. And in fact, a lot of them will sort of epigenetically or whatever take on that rhythm. So yeah, they're pretty flexible because as you can imagine, they go from Yucatan up to here, right? And so they live in these different tidal environments, so they need to have the appropriate rhythm. Anybody else? Too complicated for you?
could the females that are tricking the males that are immature, could that be practice behavior or learning behavior?
That's a good, yeah, very good point. So the lobster fishery, there's a size limit. That size limit is set at a point where 50% of the females are mature. So, right after that, so only half of them are mating to start with, okay? So after that, like 90% of them get caught. So after they molt and become big enough, they get captured and kept, right? So you can imagine there's a lot of pressure on them to reach sexual maturity smaller, right? And that pressure's only been on, for 30, 40, 50 years, you know what I mean? So I think that part of this behavior is a combination of the two clocks, one for growth and everything, and one for maturity, are a little out of sync. So they're gaining the ability to be able to have sex before they gain the ability to do it successfully. And I think it's kept in the system because it does give them practice. So their next time, for sure, they'll be good at it, right? And because it's to their benefit, turns out, right? So I think it's being pushed by a number of pressures to be smaller and smaller size at maturity. Yes?
If someone gave you $1 million and 3 more years to do research, what would you do?
Tell them to go. No. Funny Josh should ask that question, because Josh and Jason and Ben and I just submitted a grant, but it wasn't quite $3 million, but it was for 2 more years. Because we had to. Yeah, there's some, I don't know. God.
One thing.
There's one thing. I was telling these guys, I need no money for this. No, I'll tell you what I'll do. Right? You go back to the mesocosm, okay? You rebuild the mesocosm. You put these devices on these guys. You put some cameras down there, and so you learn exactly why they're going, where they're going, when they're going, right? And then I'm trying to figure out what the mechanism is. Is it path integration, whatever it is? And I have a friend who builds robots, and then we were talking about this before. It's a lobster robot. So you program the robot with the rules that you think they're using, and you put them out in the same habitat, and you ask them, can you walk away and come back? How simple is that? Absolutely.
All right, what area of technology do you think needs to be advanced the most to help answer some of these unanswered questions? And batteries cannot be the answer.
Easy. Underwater GPS. So frustrating. So frustrating. Like these guys who are out in the air have it so easy, right? Put your cell phone on an animal, let them go. But underwater, you can't get GPS underwater. So all these sort of movement things we do and stuff, you can't. And obviously, you can't stay underwater yourself to watch them. So the only way you can watch them is remotely. But we don't quite know what's going on. So I think that's going to be a huge deal. Yeah.
What's your favorite thing you ever invented?
Favorite thing I ever invented. Might be some stuff at home, but I mean, the one that's yielded the most stuff is this guy, my favorite. Which, by the way, is this big now. Yeah. Right. I like that a lot. Even though I got like 0 out of, I'd have to say that thing with the horseshoe crab and the eye cap and the running wheel. That combination was like we had to invent every step of that process. Right, that took a while. I like that a lot. Yeah.
What's the most expensive / best thing you and your grad students have broken?
Wow. Well, Dave will tell you about some bulk problems I've had, right, that were expensive. One of my famous accidents, right, is with this thing, LTV, where when we pulled it up on board, Steve Drury and I went there, I lacerated my body with it. I was bleeding profusely, and so I kind of took off my shirt and wrapped it on the wound. And then when we got back to shore, I drove to the hospital. I don't know why Steve didn't come. To this day, I don't know why Steve didn't come. So I drive to the hospital, and I don't have a shirt on, right, because I'm in a hurry. And so I take the shirt off of my legs and put it on. Right? So then you walk into the hospital, and people are like, "Oh my God!"
You get
Either
seen first?
a zombie, or I need some help immediately, which is a good thing because they helped me immediately. I did once need to dry some very expensive cuvettes, and I was, hard to believe, in a hurry. And so I opened up the centrifuge and used the wind from the centrifuge to dry them, at which time they all blew out and into the centrifuge, and it was all over. I did flood virtually every lab I've been in from where it's all. I used to be up on the second floor. Remember when I flooded a bunch of labs downstairs. So then they moved me down a floor. Then I flooded the person below me. So that's probably when I caused the most damage with my floods.
Fire.
Fire, yeah. Hard to believe. I once set this on fire. It was underwater. We brought it up and opened up, and smoke's coming, billowing out, and everything inside melted. Like, how did that happen? Yeah, we had a few. Yeah.
Wait, how many of your specimens have you eaten?
What's 25 times 40? Yeah. But they're all like disposed of appropriately. Right, Josh? That's a legitimate way to get rid of a research.
It's in your permit, right?
Yeah, it's in my permit. Right? And I've never hurt a vertebrate or treated them any poorly at all, right? Yeah. That's always fun. Like those fish were like perfectly happy.
Include grad students?
That's your grad student. Worst thing I've ever done to a grad student. I don't know. No. Yeah. No. So yeah, I've eaten a lot. Although I gotta say, down at Woods Hole every year I used to have a party at the end of summer, and it was bring your lab dish. Right. So people were supposed to bring a dish made from there. I'm working on horseshoe crabs. So I got nothing. So I had to make a dish out of a horseshoe crab and fill it with shrimp. Well, after working with horseshoe crabs, you can imagine why I moved on to salmon for a while, and lobster, and nudibranchs, which we once lost a bet. We had we didn't have a lot of money, so our basketball pool that year was whoever loses eats a melody, because they're known to be aversive.
Smell great though.
They release a chemical that prevents things from eating. We had no idea what it was. So I lost. And it wasn't bad. So now I haven't eaten another one since.
So you work with fish, crustaceans, horseshoe crabs,
Nudibranchs.
any animal or plant that you wish that you could work with.
Leech. Leech. I almost published a leech paper because the nervous system is exquisite. But if I were to go back in time and start over again, or maybe get a $3 million grant, I'd go either octopus or brittlestone. Because those fascinate me. And I'm sure there's nothing that I can figure out about them, but they're just to have them in the lab. Those are two that I work with. And I've also published a paper on an extinct species. Right? That contacted us about horseshoe crab fossils and trying to figure out what to eat. Jason and Ben had invented this pressure sensor, so we came over here and we measured how the force of biting of a live horseshoe crab, and then he was able to sort of transcribe that into what the fossils might have been able to eat. Right? So that was the easiest paper I ever found.
Yeah, Russell was here about 25 minutes. We got the data he needed, and then we sort of looked at each other and went, well, now what? All right, I guess we're going on a tourist trip.
Anything else? Luke, you got a question?
Meet me halfway.
I think Nathan might have one.
Don't make us come to the front.
What's going on? This kid will keep you busy for a while. Yes.
So obviously technology has changed quite a bit over these 40 years, and that has influenced your approach. But then from a non-technological standpoint, how do you feel like you as a scientist, because that's who you were 40 years ago versus now? So what's kind of some of the biggest differences?
You know, sorry, I'll try to close. It's hard to believe how totally incompetent I was now, then versus now. You know what I mean? I was just raw, I had nothing, right? I think the biggest improvement is now I can write clearly. I know it sounds stupid, but you know, it takes a lot of practice. So I think I can write clearly, and I think I can see the big picture. I can see the questions that are important, and then you have to take the next one, and I can see that it's what things are possible to do to get at this question. Some things are huge, but you know, what am I going to do? I think one of the things I've also had some success is other than being hyperactive and having no attention span is flexibility. So I didn't come in here with the ideas I'm going to work with horseshoe crabs the rest of my life. I see opportunities and I say, okay, now I can get money to do this, and now this needs to be done, and so on and so forth. And then don't tell anybody, and then get the money and do weird stuff. So you kind of do the stuff you're meant to do, but you know, kind of like we did, right? Like Nathan had a grant, had some leftover money. What are we going to do with all these sonar tags? We'll track some donor crabs. He had no intention of doing that when he came, but now it's kind of fun. It's good stuff. So I think the flexibility to be able to what? Oh, the exit sign.
He wants to exit.
Right. So I think that flexibility is good to sort of. You know, and then take horseshoe crabs, for example. When I started working on horseshoe crabs, you're not going to get money to work on horseshoe crabs that much, right? But then people started realizing they're valuable because of their blood. At that point, I knew enough about them that I could then ask for money to say, I can tell you how bleeding them affects them because I already have the background. So it's taking advantage of opportunities. And I think one of the things that I realized very early on is no one's going to listen to you until you build up some street creds, right? And then once you build up some street cred, you can give you money to do stuff you want to do. But better than that, right? You know? Anything else? Awesome. Thanks for letting me indulge myself.